So we've got this recorded here. Okay, good. All right. So I just wanted to start off with first saying just kind of giving you an overview of how the final is made up as far as points go. And yeah, so let me just adjust this screen here so I can see you guys a little bit. Okay, that's good. All right. Yeah, so this is pretty much the makeup of the final there. You can see my iPad screen there. 90 points on the cumulative material, 60 on the new material. And you can see the breakup of multiple choice. 53 points of cumulative multiple choice, 47 points of new multiple choice, and then some hybrids there, 37 on the cumulative stuff, and 13 on the new stuff. Okay, so that gets us to the 150 points. And that is what the final is made up of. Okay, so yeah, we'll use honor lock like normal, and yeah, and you have the full 2.5 hour time block, so should be plenty of time. Okay, any other questions about just like the makeup of the final before we get into some actual reviewing of content? No? Okay. All right. Okay, well, I thought we would start out maybe with looking at the new material a little bit, looking at the kidney stuff and the fluid balance stuff. And so, yeah, let's do that. Does anybody have any specific questions about some of that material? That I know probably the couple places we can go would be looking at the nephron, different parts of the nephron, and kind of what things happen there at each part in the urine production notes, or we could jump into the regulation of urine and body fluid one with the hormones and the sympathetic nervous system and stuff. But does anybody have any questions there that we want to start with?
Can we start with the structures of the nephrons and which ones are more common?
Sure.
The differences between two?
Yeah, definitely. And that's in this first one, I think. So let's jump in there. Okay, so yeah, in the first set of notes here, in the kidneys stuff and the urinary system anatomy, we've got, yeah, this is a good slide to start with here. And this is, yeah, the one that I had already made notes on. This is a really nice image if you can kind of wrap your head around what's going on here. So if we look here, we've got the kidneys, right? What this image is zooming in on one part of the kidneys right here, right? But you can see that part of the kidneys stretches all the way from the cortex down deep into the medulla, the part that we're looking at. Okay, so the top of this would be like the very outside of the kidneys, the medulla or the cortex, right? Kind of close to the very outside, and then the bottom of this down here would be kind of down here in the deeper part of the kidney in the medulla. Okay, and then this right here is that junction right here, the cortex medulla junction, that is just kind of that kind of mid space in between. Okay, and in the much more common type of nephron is this cortical nephron. That's 85% of the nephrons. Okay, and that's this guy right here, right? That's this one. Let me actually just switch to a different color pen, maybe to make it a little more obvious what I'm writing here right now. So yeah, this cortical nephron here is the much more common type, and it's mostly in the cortex, right? It's in the outside part, so it's in this part of the kidney. It's in the far outside part that contains the peritubular capillaries. Okay, you can see we don't have that around these kinds, right? There's no capillaries around the distal and proximal tubules in the other kind of nephron. Okay, it's the loop of Henle is much smaller, right? And here's those differences there, right? Nephron length, where it is next to the cortex of the medulla junction, and then the vasa recta versus peritubular capillaries, okay? So yeah, so that's the big one there. So those ones do most of the work of reabsorption of sodium and glucose and all those things, all the stuff that would happen in the proximal and distal tubule. That all that stuff is mostly done by the cortical nephrons, okay? But then we have these other nephrons, these juxtamedullary nephrons, which just means next to the medulla, right? Or they're within the medulla, and they go much deeper, right? They've got this super long loop of Penley. They have the vasa recta around the loop of Penley to help us with that countercurrent multiplier mechanism, right? So these ones are creating this super high concentration, right? Or high osmotic pressure. They're creating a high osmotic pressure deep in the medulla of the kidney, which is all going to be set up for the collecting duct, right? So we want it to be as concentrated as possible as we go down there, because as the urine is flowing through here, the collecting duct heading out, like it'd be going this way, heading in the kidneys towards the ureters, right, towards the renal pelvis, water is being pulled out, right? And the only way water can be pulled out of the collecting duct there is if this guy did its job, the vasa recta and those long loops of Henle on the juxtamedullary nephrons, and created a strong gradient so the water can get pulled out. Okay, so that's only about 15% of the nephrons, but very important for urine concentration and saving water in the body. Okay, so those are kind of the two main types and some of the main important differences between the two. Any other clarifications on that or questions related to that?
I have a question. Why
Yeah.
do we have two different types of nephrons? I'm not sure if you covered in the notes, but why? I was just wondering why we have two different types of nephrons.
Yeah, I mean, it's just because they kind of serve different functions, right?
Okay.
The cortical ones are really important for all of the reabsorption that happens. Reabsorption of sodium, all the things we want to get back, also really important for the secretion that happens. So getting rid of hydrogen ions, getting rid of urea, all the nitrogenous waste products. Those cortical nephrons do most of that work. They do most of the pH balance for us, and they're highly specialized to do that with a very long, with kind of the longer, this portion, the proximal and distal tubules with that special capillaries around. And then the other ones are just specialized to do the other part, which is set up that gradient around the collecting ducts so we can get all the water back at the end. So they just kind of serve different functions. I don't know why we couldn't have one type that did both of those things. It's probably just not as efficient to have so many of those big long ones, because I guess we don't need so many of those big long ones to get the job done, right? So maybe those are kind of more extravagant in a way. We don't need those super giant ones. We don't need so many of them. Maybe that's part of the reason. I don't know, but it's they do have different functions.
Okay.
It's a good question. Yeah. Yeah. Anything else on those? Okay. Yeah, so yeah, so anything else there? This is also a key thing to understand and know. Oops, where did I go? The wrong way. Meant to be right there. Yeah, so this part right here, this juxtaglomerular apparatus, okay? This is a very important part of the kidneys. And we can see here that this is, if we're following, let me get back to the blue here. So if we're following the nephron over here, we go through proximal tubule, right, loop of Henle, distal tubule, and then where the distal tubule actually comes up behind the glomerulus, which is how they normally are, that's where we are right here, okay? All right, so here is that distal tubule. So this part of the distal tubule is right there, right? So it's kind of hard to get your bearings sometimes of where we're at there. But so the distal tubule of that same nephron wraps around and goes back up right behind the glomerulus, okay? And so here's those arterioles coming in, right? So there's the afferent arteriole, efferent arteriole. And so we have really kind of three structures right there that are important: the distal tubule, afferent arteriole, and efferent arteriole. And there's two different types of cells in there, right? So these cells that are surrounding the afferent arteriole, known as the granular cells, what do? They monitor blood pressure, right? So the more that blood is pushing out on those, they're sensing exactly how much blood is pushing out on those. And that's one of our primary ways of monitoring blood pressure in the body, okay? And so those are also the cells that make renin, right? So when we have a drop in blood pressure, those cells sense it, they release renin into the blood, and then we set off that whole chain of events that leads to the formation of angiotensin II, right? And that's an important thing to remember too. Renin is going to be released when we have a decrease in blood flow to the kidneys. It's sensed right there by the granular cells, and then that's going to go in, interact with a blood or with a protein that's always there, which is angiotensinogen. And let me write that out real quick. Let me just write out that chain of events here real quick. So we have decreased blood flow to kidneys. Okay, that's going to lead to renin release by those granular cells, the ones we just looked at. Okay, so we got renin release, and then renin. So then we have a angiotensinogen, and this is a plasma protein that's made by the liver, right? Always in the blood. Okay, it's going to interact with renin. Renin is an enzyme. Okay, renin is going to turn it into angiotensin 1. Okay, oops, there's no O there. Angiotensin 1. Okay, so angiotensin 1 is then going to interact with another enzyme called ACE. Oh, bless you.
Oh, shoot. Sorry.
That's okay. Scared me a little bit. Yeah, so there's another enzyme that's always in our blood called ACE, and ACE is made by the lungs. That doesn't mean that it's only in the lungs, it's just made by the cells in the lungs, okay? And then that turns angiotensin 1 into angiotensin 2, okay? And that whole chain of events is very important to know, but this all started by the juxtaglomerular apparatus and those granular cells that sense blood pressure and release renin, right? And so then the effects of angiotensin 2 are a lot. Okay, there's number one, it's a vasoconstrictor. Okay, number two, causes water retention. It does that mostly by stimulating ADH to be released. Also causes sodium reabsorption. Does that mostly by causing aldosterone to be released. Okay, and then we also talked about how it increases thirst. Okay, and I think I might have said it. The image I had in the notes didn't have this directly spelled out, but I probably said it. It also directly increases our sympathetic nervous system activity. Okay, so there's kind of these 5 effects of it, and all of these would come together to increase blood pressure, right? So that all gets back to the initial stimulator of this whole process, which was low blood pressure in the kidneys, right? So then we get all of these things. We get more sodium, more water, we drink more, we vasoconstrict our blood vessels, and we get sympathetic nervous system activation, which vasoconstricts more. All those things lead to more water being saved, more salt being saved, more sodium being saved, and then that increase in blood pressure. Okay. I'm going to go back to the juxtaglomerular apparatus here in a sec, but any questions on that process? Because that's a very important thing from this section for sure.
I have a question about that portion.
Go ahead, Trey.
So is aldosterone then like triggered to be released after angiotensin 2?
Yes, that's one of the main stimuli for aldosterone release is angiotensin 2.
Okay.
Yeah, so we also have that high potassium in the blood will cause aldosterone release. And we also have the whole chain of events with ACTH being released from the pituitary gland that causes aldosterone release. But angiotensin 2 is another mechanism of causing aldosterone release. So then the other thing there that I didn't say is that we increase potassium secretion, right? So we get rid of potassium with aldosterone too, right? Because when aldosterone is acting on the distal tubule, it causes the distal tubule to pull sodium out of the urine back to the body, and then put more potassium into the blood. Into the urine to get rid of. Okay. Any other questions on that? Okay, let's go back here. Excuse me. So that was just the first part of the juxtaglomerular apparatus. That was the granular cells. Okay, but then we also have these macula densa cells over here. Okay, and you can see they're not doing anything with the blood, right? Those are right here. Those are monitoring something in the filtrate, right? In the urine, in the tubes of the kidneys. So they're monitoring solute concentration in the kidneys. Okay, and so that's an important piece too of what this juxtaglomerular apparatus does. Okay, so that's kind of going to help us to dictate the whole. It's going to help us to dictate the concentration of the urine and help the body to decide how much sodium to pull back, how much potassium to put back in. Okay, so that's a very important part of the kidneys. It kind of has a regulatory mechanism. Okay, questions on those? Okay, so then, yeah, this is another good slide to look at your question, Becky, about the different types of nephrons. Well, I think it was yours, Becky's, and Kayla both asked about this, to look at those different types of nephrons. And this is just a good one to look at. So just take a look here with me. So here's a little cortical nephron, right? Here's a juxtamedullary nephron, right? Here, this shows the minor calyx, the major calyx, going down into the renal pelvis down here, right? Which would then lead to the ureters. So this kind of shows you everything all together, right? This would be a nice way to really see kind of everything. And then this shows you the whole pathway of filtrate from Bowman's capsule all the way to the ureter. And so, yeah, that's just a nice figure to look at with that. Okay. All right, so let's see. Other questions here on that? I can definitely hop into the urine production notes, which I'll go ahead and hop in there quickly and show you a couple things there. Okay. So a couple key aspects of this are so probably if we jump here to these this part of the notes where you guys were to kind of recap what happened in each section, this would be a really key thing to know and to understand. Okay, so make sure you have this part memorized, right? So what happens in the proximal tubule, what happens in the distal tubule, what happens in the ascending limb versus the descending limb of the loop of Henle, and then what happens in the collecting duct here. Okay, so all of that is really important just to make sure you understand that. Okay, and yeah, we can I'll just show this one here. This whole countercurrent multiplier mechanism thing is important, and really just the basics that you need to know about it are one thing, we have fluid flowing in opposite directions in little tubes that are right next to each other, right? So fluid flowing down here, right, up here, okay? That kind of creates this little whirlpool effect around the kidney, or around the little distal tube, or around the loop of Henle, sorry. We get that little whirlpool effect. That helps to pull water out here, but it also helps in the process of pulling sodium out here. So all in all, that gives us more ability to pull water out on this side, on the left side, or the descending side, more ability to pour sodium out, which is going to help us to create a very, like it says here, very concentrated interstitial space down there below or in the deepest part of the medulla. Okay, so then when we look at this, this here, this is where that vasa recta comes in, because when the water comes out, we don't want to just let it fill up out here in the interstitial space, because then that would dilute that space, right? So immediately the vasa recta picks up that water and sends it back out, so we can continue that process, right? Same thing with the sodium. We want to pick up that sodium, keep it moving. And as you can see here, the most concentrated space is the deepest part of the nephron. Least concentrated up here, 300 milliosmoles versus 1200 milliosmoles. So the deeper we get, the more concentrated we get. Okay. Any questions on this set of notes that you want me to talk about? Lauren's asking, do we need to know the micturition reflex on the study guide? But I don't. Something. Yeah, the micturition reflex is the urination thing. It's on here. So right here. Yeah. So this is the micturition reflex. You do need to know that. So it's very similar to the defecation reflex, where we have in the rectum, when the feces fills up, it stretches out. Stretch receptor sends a signal up to the spinal cord. That spinal cord sends a signal right back, which relaxes the sphincters, right? There's that internal sphincter that gets relaxed. And then it contracts the rectum to push the feces out, right? So same thing here with the urination process or micturition reflex. The bladder stretches. Stretch receptors send that signal back to the spinal cord. The spinal cord sends two signals down, one to contract the bladder and the ureters, right? So squeeze it out, right? Squeeze it out, but then also to relax or inhibit that internal urethral sphincter, right? So we can see that right here. So here's the internal one on males versus females. And like I said in the notes, there's a little bit of space in here, right? So that's why it actually can kind of feel like you do almost pee because it kind of can get into that little space between the two sphincters, right? But you've got control of this external sphincter voluntarily. Okay, so that's the micturition reflex. You definitely need to know that. Yeah. You're welcome, Lon. Other questions on this set of notes here?
Can we go over the glomerular filtration rate?
Yeah, for sure. Okay. So yeah, so with this, it's pretty much just a way to measure how much filtration is happening, right? So we have the filtration really occurs by the blood going into that afferent arteriole, right, of the glomerulus. Then it goes into that whole kind of complicated set of capillaries in the glomerulus, and then it's kind of spinning around in there, and fluid is getting pushed out as it's in there, right? And that's filtration, right? All the fluid that gets pushed out of the glomerulus into the kidneys, in the kidney tubular system there, in that Bowman's capsule, that's filtration, right? So we send about 1.2 liters per minute of blood to the kidneys. That means that's how much is going through that afferent and efferent arteriole. Okay, of that 1.2 liters, only about 120 to 125 mils per minute is actually pushed out, right? So most of it stays in the blood, in the actual arterioles, right? But some of it does get squeezed out through those capillaries, and that's the filtration. Okay, so then when we when we look at this, so here we'll skip to the next part. So a GFR, glomerular filtration rate, is exactly that. It's 120 mils per minute, right? Because it's how much fluid is getting pushed out of the blood and into that tubular system as the blood goes through the afferent and efferent arterioles, through that glomerulus, how much of it actually gets squeezed out, right? So that's GFR. And when we, yeah, we can kind of briefly touch on it, but it's definitely important to understand that if the whole clearance, renal clearance concept, right, the RC, we used to have you guys calculate it. I won't have you do it this time, but it is important to understand that so renal clearance means how much of a certain substance is the kidneys putting into the urine, okay? And so if we have a renal clearance, oops, let me go back here. Yeah, so if renal clearance is greater than GFR, okay, so we just said GFR is typically 120, okay, so if we have our GFR of 120, but we have a renal clearance of 300, okay, then that's gonna be something that the body's trying to get rid of, right? Because whatever amount of that substance was in the blood when the filtrate was created during filtration, that same amount went into the tubular system. So if we got more than that, that means as the filtrate was going through the kidneys, we were actively putting more of that in there. Okay, so these would be like waste products. Okay, so these would be hydrogen ions. These could be Tylenol, right? Any medications or waste products. Things that your body is actively trying to get rid of. Okay, so that's if renal clearance is greater than GFR. Okay, let me switch over to this so I have a little more room to write. So renal clearance greater than GFR is going to be waste products or toxins. Okay, but if renal clearance equals GFR, okay, so if we had our 120 GFR and we had our 120 renal clearance, these are, and so what we would say about these waste products and toxins, we say that they would be freely filtered. That means that they go with the plasma, and as it gets pushed out of the glomerulus into the tubules, so freely filtered and secreted. Okay, that's the key word. It's going to be there. They're freely filtered and secreted, meaning the body puts more of it in there. Okay, if renal clearance equals GFR, these are going to be things that are, again, I'm just going to do FF for freely filtered because that's always the case, okay? And not secreted, but also not reabsorbed. Okay, so it's like whatever amount of the substance was in the blood got pushed into the filtrate, and the kidneys just left it there. They didn't try to pull it back. They didn't try to add more of it. It just stayed there, right? And the two that we talked about were inulin and creatinine. Okay, and those ones are just they're really, yeah, one's a starch, one is a waste product of breaking down creatine, but both of them are good measures of kidney function. Because we know if we look at the renal clearance of those, that's going to match GFR. So if we just measure those, we say, oh, okay, that gives us a good idea of how well the kidneys are working. Okay, and then lastly, if renal clearance is less than GFR. Okay, so if our GFR again is our 120, we'll put this up here just to complete it, and then renal clearance is only 50. Okay, these would be things that again are freely filtered, okay, and reabsorbed, right? Because we put them in there, right? They went in with the filtrate when we had that filtration, and then the body said, nope, actually I want to get that back, right? I don't want to let you waste that, so it's going to pull it back, right? So these would be nutrients like glucose, amino acids, and sodium. Okay, those were the three main examples that I think that I gave in the notes. Okay, so that's an important concept to understand. Okay, other questions on that? Okay.
That was
Go
it
ahead.
for me.
Okay. Okay. Thank you. All right. So let's take a quick look here at the regulation of urine and body fluid. And that's an important one. Okay. So there's a lot here in this set of notes, but most of it is stuff that that you guys have already seen, right? We've already studied ADH. We've already studied aldosterone. We haven't talked too much about ANP, atrial natriuretic peptide, and we've already talked about the sympathetic nervous system, renin, angiotensin 2, all of these things, right? We've talked about those, but this set of notes just kind of brings all those together and shows you how they all work together, right? And that's the key part of this. Okay, so take you to a couple things. This would be a good thing to kind of make sure you understand here, right? So which of the following factors leads to an increase in blood pressure? Okay, so I went through that in the notes. Make sure you're good with that. And then these ones here are very important to understand, right? So all four of these last four slides are very important. Okay, so it's called the integrated response, and there's 4 different scenarios, right? So scenario number 1 was high osmolarity, which would be like high concentration in the blood or in the extracellular fluid. Okay, so meaning like low water, high salt, right? So what do we do with that low water, high salt? Well, we that first of all, that's sensed by those osmoreceptors in the hypothalamus and the JGA, that juxtaglomerular apparatus. Okay, and what were those cell types called? It wasn't the granular cells. What were the other ones? Somebody tell me.
Macula densa.
There we go, those macula densa cells. Thank you. So that's where these come in. Okay, so those are sensing that that osmolarity. Okay, so then look, the thing here with the osmolarity disruptions, ADH and renin move in the opposite direction every time. Okay, so we have if we have low water, high salt like we do here, then we got to increase ADH, right? ADH's primary job is to increase water reabsorption, right? And that's the main thing. It's also going to make you thirsty. Okay, but so we're going to bring more water back, but we've already got enough sodium, right? So we don't want to bring more sodium back. So we actually want to decrease renin, even though renin has also has the effect of water retention. It's much stronger of an effect on sodium. Okay, so we want to think of ADH as the water one, renin, the whole renin, angiotensin, aldosterone system as the sodium one. Okay, so this is going to decrease sodium reabsorption. Right? So the final result of this would be decreased osmolarity. Why? Because we have an increase in water, increase water into the body, right? And then we're going to increase sodium out of the body. Maybe that's a weird way to say it. We could just say in for both of them, but increase water in and then decrease sodium in, right? Or just but we are actively sending more of it out. Okay, so that's going to lead to that decrease in osmolarity. Okay, the extracellular food is going to become less concentrated, more dilute. Okay, that one makes sense. Questions on that one?
So this isn't like a blood pressure. We're not talking about blood pressure here, just the osmolarity.
Just osmolarity on this first one. Yep. Mm-hmm. And when osmolarity is the problem, then ADH and renin do the opposite thing. That's a key thing to remember. Okay, because it's not that salt and water are the problem here. It's one or the other, right? In this case, with high osmolarity, it's that we don't have enough water and we have too much salt. Okay, so we got to do the opposite thing with ADH and renin. Now, when we look at the next one, it's the opposite. It's low osmolarity. Okay, so low concentration. Okay, and hi. You guys want to say hi to Eve real quick? You say hi, miss, to everybody. What is that? Miss Mason's there somewhere too. She doesn't have her camera on there. Okay. All right, Miss. I'm gonna keep working with him, okay? Can you say bye to him now? Say bye, everybody.
That's you.
That's me. Yep, I'm on there too. It's in my class.
Oh, can I say hi?
Okay, say hi real quick. Here's Soren. Still in his jammies. Eve's trying to block Soren's view, of course. Okay, guys. See you later. All right. That's work at home. It's fun. Okay.
Dr. Sawyer, you're on mute.
Say that again. Can
You're
you
on
hear me?
mute for a second.
Okay, yeah, sorry. I think my headphones were just kicking back in. Thank you. Okay, so we got the opposite problem here, low osmolarity, which really means too much water, right? And not enough sodium. Okay, that's really the problem here. So then the way to fix that is lower the one that brings more water in and raise the one that brings more salt in, right? So this decreases water retention, and this increases sodium reabsorption. Okay, so the end result is increased osmolarity. Why? Because we have less water in and more sodium in. Okay, so that's the deal when osmolarity is the issue. And that's a little bit of a simplified version of what I gave you before, but that's the bottom line. Okay, questions on those before we go on to the volume ones? Okay, so then if we have a volume issue, which is just total volume of blood, this would be sensed by baroreceptors, right? So pressure receptors. We have these in the atria, big veins, carotid sinuses, aortic arch, and then also those cells in the juxtaglomerular apparatus, those other ones, not the macula densa, but what were the other ones called? Somebody tell me. Come on, guys. Somebody tell me. You're all muted. You got to unmute yourself. Okay. Yeah,
Yeah.
Jasmine's saying granular cells. Thank you. Those are the ones sensing blood pressure there. Okay. So that goes back to that, connects back to that too. That's important, right? So, okay. So now we got 4 things going on here. We'll notice that ADH and renin go together in this case, and that they're going to go along with the sympathetic nervous system too. Okay. So too much blood volume, right? So we want to get rid of water, and we want to get rid of salt because that's going to help us get rid of water. Okay. So we got to get rid of fluid. So we're going to decrease sympathetic tone. What does that do? That vasodilates. So that doesn't help us get rid of fluid, but that does not that vasodilation piece there, but that does help us to decrease blood pressure. Okay, and then also we increase atrial natriuretic peptide, which pretty much what that does is the exact opposite of ADH and renin. So that's going to kind of block ADH. It's going to block aldosterone. Okay, so those things together are going to decrease water retention and decrease sodium retention. Okay. Yeah, so, and then that's the same thing this is going to do, right? So decrease in ADH will decrease water retention, decrease in renin will decrease sodium retention. So the final result of this is a decrease in blood volume. Okay, and then the other thing that this sympathetic tone does is it's going to vasodilate dilate the afferent arteriole. In that nephron, right? So if we do that, that's going to actually increase our GFR, right? Because that's sending more blood into the kidneys, creating more filtrate. So if we increase our GFR, that's going to eventually cause more urine output, right? So we have a decrease in blood volume because we have less, we could say it this way, we have more water out, more sodium out. Okay. All right, questions on that one? Okay. All right, and then the other one is just going to be the opposite. I don't necessarily need to run through it because it's just going to be pretty much the exact opposite. Instead of vasodilating the afferent arteriole in the nephron, we're going to vasoconstrict it, which will lead to a decrease in GFR. We're going to decrease HO natriuretic peptide, which will allow for ADH and aldosterone to do their job. We're going to increase ADH and aldosterone. We're going to hold on to more water, hold on to more salt, increase blood volume. We'll end up with more water in, more sodium in. Okay, so that's an important piece of that. Questions on that? And this is kind of showing that piece here in the changes in the afferent arterial diameter with the sympathetic nervous system. Vasoconstrict it, that's going to decrease GFR. Vasodilate it, we're going to increase GFR, create more filtrate. Okay. Yeah, so I'm inclined to move on to any review stuff that you guys might be, or cumulative material you might have questions on. But if you have any other questions on that stuff. Sophie's asking the A&P recap. Yeah, we can do that. That's back here. Where are we at? Peptide. Yeah, okay. Yeah, so right here. All right, so AMP recap. Okay, so what effect does AMP have on ADH? So atrial natriuretic peptide is stimulated by stretching out the atria because there's too much blood, okay? This is common in heart failure patients to see high levels of AMP with congestive heart failure. Okay, so ADH, it's gonna block it, right? Block ADH, which is going to decrease water retention. Okay, it's going to block renin, which is going to lead to decrease in angiotensin 2, which leads to a decrease in ADH and aldosterone. Okay, so and then it also is going to block aldosterone. So what does that mean? That means less sodium reabsorption. Okay, effect on arterioles is it's going to cause, it's going to cause vasodilation, as it shows there, right? So it's going to vasodilate. That would be didn't spell that. I keep forgetting that L there. So the vasodilation is caused by that decrease in angiotensin 2, right? Because blocking renin release. Okay, and then the overall effect would be to decrease blood pressure. Okay, it would be to decrease water in the body, decrease sodium in the body, and vasodilate. Okay. Good, Sophie? Any other questions from you or anybody else on that? Okay. You're welcome. Okay. All right. Any other questions on the stuff with the urinary system before we do any review material? the fluid balance and urine clearance one. We kind of already went over some of that with the renal clearance stuff. But then the other piece is just kind of knowing kind of where most of the water in the body is, and being able to, yeah, kind of just account for like where is most of the body's water content, and some things like that. So go ahead. We got a question on that? No. Trying to get here. Yeah, here we go. Yeah, so this just kind of looks at this graph. This chart is definitely important to see kind of, you don't have to memorize the exact numbers, but you want to know in general where most of the water in the body is.
80, 20, 20.
Somebody got a question there? Might just want to mute your mic if you're talking to somebody else. Okay, yeah, and then the rest of it is really just about the renal clearance stuff that we've already talked about. So, okay. All right, so what questions do you guys have on the cumulative material? Is there anything you'd like me to dive into there to kind of refresh your memory on? I know there's a lot of options, a lot of choices.
I have a question about section 5.
Okay.
So I was looking through the study guide, and under the carbon dioxide and control of ventilation, one of the things that you mentioned were going to be on the test is knowing the details of the Herring-Brewer reflex, and I couldn't find that through the notes.
Yeah, thanks. No, that's a carryover that we took out this year, and I forgot to take it out of the study guide. Sorry about that.
Okay.
Yeah. Good clarification. Yep, don't need to know that one. What else?
In section 5, could we go over the carbohydrate and protein and lipid digestion and absorption again?
Yeah, for sure. Yeah, so that would be here. No. Yeah, this one here. Yeah, so the big thing to understand, just as you had to understand before, was the kind of being able to track where digestion of each of those starts and ends, and digestion would be breaking it down, right? And then secondly, how absorption happens, and then what happens to it after that, right? So so this stuff at the these notes at the end show most of it. Okay, so for the wait, I went the wrong way. Sorry, went the wrong way. Okay, so carbohydrate digestion and absorption. So anytime we see an amylase, or we know that that's something to break down carbohydrates, right? And that's going to start in the mouth, right? Because we have that salivary amylase, right? So that's where carbohydrate digestion begins, is in the mouth. And so that starts breaking down long polysaccharide chains into shorter polysaccharide chains, okay? And maybe even the short polysaccharide chains that you ate are getting broken down into some disaccharides. That's why some of that bread or something like that might turn a little bit sweet in your mouth, okay? If you let it sit there, okay? And then once it gets into the stomach, not a whole lot happens in the stomach, as we saw in the lab, because what happens is the pH of the stomach just destroys that pancreas or that salivary amylase, okay? But then we get through the stomach into the small intestine, and we have two big ones working there, which are, change my pen color again, which are going to be pancreatic amylase. Okay, so that's the first one. That's going to be breaking down polysaccharides into disaccharides. Okay, so those shorter chains of polysaccharides are now getting broken into disaccharides. And then lastly, we have brush border enzymes designed specifically for each of those disaccharides to turn those into monosaccharides. And then the monosaccharides are absorbed via co-transport. And so a couple people got mixed up on the exam on this one. Co-transport is a general term meaning that two things are transported together. Could be the same way, could be the opposite way. It doesn't necessarily tell you whether that's active or passive transport, but in this case, we're talking about secondary active transport of monosaccharides, okay? Because there's, like it says here, there's always gonna be more, typically always gonna be more monosaccharides inside of cells than there are outside of cells. So in order to get more of them to come in, this way, we need energy to do that because they're going against their gradient, right? So but sodium is the opposite, right? Sodium, we've always got more on the outside of cells than we do on the inside. So sodium has a gradient, so it can passively blow through here with the help of a protein. And when it does that, that protein uses the energy of the sodium gradient to pull along a glucose as well. So this is not just co-transport, but it's also secondary active transport, right? So using energy, but not using ATP. Primary active transport would be using ATP. We're using secondary, using the gradient of something else. Okay, so that's a key part. And then down here, we see this, our old friend, the sodium potassium pump, right? That's primary active transport, right? That's using ATP to pump sodium back out and potassium back in, okay? Why do we need to do that? Well, we need to do that because if we just let all this sodium that's being used for secondary active transport to get glucose in just build up in cells, then we would no longer have a gradient for sodium, right? So we got to get that sodium out in order to maintain that gradient. And that's where the sodium potassium pump comes in. Okay, so that's the carbohydrate one. Any questions on that one? Okay, and then the protein one is somewhat similar, but a little bit different. We don't start any protein digestion until we get to the stomach, okay? Protein digestion begins in the stomach with the low pH. The low pH denatures some of those long protein chains, right? Starts getting them to unfold. And then we have pepsin in the stomach, right? Pepsin is released in the form of pepsinogen, but then when it hits that stomach acid, that turns it into pepsin, which is an active enzyme, which is going to break down some of those super long polypeptide chains, chains of amino acids into shorter chains of amino acids, right? So that's all happening in the stomach. Once it gets into the small intestine, now that's where we are here. We have pancreatic proteases, right? And there's a bunch of different ones because there's so many different types of proteins that require different enzymes to break them down. So we've got all these, so trypsin, chymotrypsin, carboxypeptidase, okay? Now these are breaking down those peptides that got a little bit smaller in the stomach to even smaller chains of peptides, okay? That still just means chains of amino acids, okay? So now we got these short chains of amino acids, and then they interact with brush border proteolytic, right? That just means breaking down proteins, enzymes, and finally breaks those down back into their individual amino acids, and they're absorbed the exact same way shown right here, right? Using that gradient of sodium to help us drive in free amino acids, using the sodium potassium pump there to maintain that sodium gradient, and then it goes into the blood. Okay, and that was the same thing with carbohydrates. Those monosaccharides go through the intestinal cells into the blood, right, into capillaries. Okay, so that's key there. The one thing I'll say, the lipid one is much more, a little bit longer, right? But we do have a gastric lipase. That was one point of confusion because there was one point in the notes I said before that when we talked about the stomach that said the only thing digested here is protein. There should be a little caveat there that we do have gastric lipase that starts to break down fat. So that's where fat digestion begins, is in the stomach. And then we get bile salts to emulsify the fats, which just means break them up, right? Separate them from the others. Pancreatic lipase breaks those down into smaller fat droplets, right? We get them carried in micelles. Micelles drop those off at the intestinal cells. Inside the intestinal cells, they get surrounded by proteins, turned into chylomicrons. Those chylomicrons then get absorbed into the lymphatic system, not into the blood. A lot of people mix that part up on the exam too. Lipids don't go right into the blood, they go into the lymphatic system. Then eventually back to the big veins, dumped into the liver, converted into cholesterol, different cholesterols, LDL, HDL, etc. Okay, so that's a quick shot of the lipid one. Definitely important processes to get and understand. All right, what else, guys?
Can we go over the menstrual cycle phases and
Yes, absolutely. Okay, where was that? Section 3 or 4? It was 3, huh?
yeah, section 3.
Okay, so yeah, probably the best place to start is here, and then we'll go to each phase. So this female sex hormone loop is an important one to understand. So we're starting with the hypothalamus, right? I didn't give you the name of this hormone. You'd have to know the name of this hormone, but it's a hypothalamic hormone. Let me go to blue here. Hypothalamic hormone, right, goes to the anterior pituitary, causes two hormones to be released, right? It's that FSH and LH, FSH, LH. LH goes to the thecal cells, right? FSH goes to the granulosa cells. So two different, that's an important thing just to know, LH, thecal cells, FSH, granulosa cells. Okay, those thecal cells are going to be stimulated to release androgens, which eventually will turn into estrogen. Okay, FSH, it's causing those granulosa cells to grow and multiply, right? So that's causing the growth of the follicle, right? Granulosa cells also release inhibin. Okay, the result of all of this is we get a lot of estrogens, right? Estrogens released directly from the follicle and these androgens from the thecal cells. Okay, at first, in most phases of the menstrual cycle, except right before ovulation, this level of estrogen is negative feedback on the hypothalamus and the pituitary. Okay, so that kind of regulates the amount of FSH and LH being released, right? So that's the normal thing. But then at some point, when the follicle gets to its biggest point, where it looks like a little clown in this one for some reason, it's got the little nose there and the eyes, the big mouth, but when it gets so big, that means we're releasing a ton of estrogen, right? So the estrogen levels get so high that instead of negative feedback that we saw over here, this turns into positive feedback. Okay, there's some tipping point that occurs here. So then instead of regulating and lowering the amount of estrogen released, it goes the opposite way. We get a huge rush of both LH and FSH, but FSH isn't the important one at this point. LH is the big one, that we get that surge of luteinizing hormone. And this is happening right around, oops, sorry, I just turned my iPad off. We're back. There we go. So this is happening right around ovulation, right? So that LH surge is what causes the follicle to rupture, right? Causes that follicle to rupture, the egg can come out. That's ovulation, obviously. And then, and then everything drops back down. Estrogen levels drop back down, progesterone levels now start to rise, okay? We still have pretty high estrogen, but it's starting to drop. But this is really where we see progesterone levels kick in, and that's all due to the corpus luteum now, right? The corpus luteum is just this follicle now post-egg release, right? It's just a follicle that doesn't have the egg in it anymore. It's now this kind of endocrine organ that's releasing progesterone, estrogen, and inhibin, but progesterone is kind of the dominant one here. Okay, so this is during the luteal phase of the menstrual cycle, right? And that's the most consistent in time as far as the menstrual cycle goes. It's typically almost always 2 weeks. And that's because of the life of the corpus luteum is somewhat consistent. Okay, so that's kind of the hormone loop. We'll go to each of the phases here in a sec and tie that together with that. But questions on those pieces first? Okay. All right. So, so the first phase is the menstrual phase. It's just the easiest one to know because it's the first sign of blood, right? All the other phases, we don't really know what's going on unless we're actually taking measures of hormones, different things. But as soon as blood shows up, then we know that's the start of the menstrual phase, and that's why it starts there. Okay. So this is days 1 to 5, right? First sign of blood. At this point, the corpus luteum dies. It's the end of that 2-week luteal phase, right? So that progesterone levels drop because that's where the corpus luteum was doing, was releasing progesterone, which was maintaining the endometrium. It was keeping it alive in case implantation happened, right? Okay, so the endometrium dies because progesterone levels dropped. So all the blood vessels, mucus, and membrane all falls off. This is the discharge. Okay. Now we see low levels of FSH and LH in this phase, but they're starting to rise a little bit because progesterone was inhibiting the release of those. Okay, so that's the menstrual phase, 1 through 5. Next, 5 through 13, leading us right up to ovulation. This is what's called the proliferative phase. What's proliferating? It's the follicle, right? The follicle is proliferating. This is when FSH is the key player, right? It's doing its big job of causing the follicle to grow, right? The follicle is growing because we're getting all that follicle stimulating hormone. Okay, the more it grows, the more estrogen is released because it's released by the follicle. Okay, so at first we get negative feedback, right? That was back to that part, and then that helps keep these levels constant. So the endometrium is growing and the follicle is growing. So it's not just the follicle that's growing, but it's also the endometrium because estrogen is causing that. Okay, and then that leads us right up to day 14, which would be ovulation. Again, this takes us right back to that tipping point of so much estrogen because the follicle is so big, turns it into positive feedback, leads to that LH surge. Again, that means luteinizing hormone, right? The hormone that turns the follicle into the corpus luteum, and that stimulates ovulation. Okay, ovulation happens. Now estrogen levels immediately begin to drop. Okay, and then we move into the 2-week phase. That's the most consistent again, luteal phase, or it could also be called the secretory phase, depending on if we're focusing on the follicle or on the endometrium. Okay, so the corpus luteum is releasing high levels of progesterone. Progesterone is maintaining the endometrium, right? It's keeping the endometrium alive, and it stimulates secretion from glands, right? So all those glands, that's the secretory phase. This creates a mucus plug. That's all kind of preparation for pregnancy to happen, if possible, or if it does happen. Okay, lots of increase in blood flow, lots of extra blood vessels being grown here. This is where the blood comes from eventually. And then in this phase, we're having low levels of GnRH, which is the one you don't have to know. That's the hypothalamic one, FSH and LH, because it's being inhibited by progesterone. Okay, and that takes us right back to the beginning. I'll just go to the end here so we can see all those together. So there we go. Yeah. So that kind of shows that. So definitely a good image to study and understand and connect that back to the hormone loop that we started with here. And again, this is showing the uterus here, right? This is showing that growth in the endometrium, adding blood vessels, adding mucus membrane, right? And that's all happening during that proliferative phase. It's starting during the proliferative phase, right? That's when the follicle gets to its biggest because it's proliferating. But the endometrium is also proliferating. Okay, ovulation happens at day 14, right with that LH spike. Right, ovulation occurs. Now we've got corpus luteum, and now we see progesterone levels spike. Right, that's where we really see progesterone levels spike is when that corpus luteum is there, starting to degenerate. So progesterone starting to drop. Okay, when it drops enough to be completely degenerated, then we start back over at the beginning, and that's what initiates the menstruation, is because the progesterone level is low enough for the endometrium to die, not be maintained, and that's what we see here. Okay. All right. Questions on that? Yeah, you're welcome, Jamie. How are we doing on time? Yeah, we've still got another 20 minutes if we want to keep going. Some other questions. I can go till 10:30.
Did you go over the negative feedback loops in section 2?
Yeah, just in general, or they're just kind of general what that concept means, or they're
Yeah,
specific?
just like in general, like some of the general ones we would need to know, like the ones that you were talking about on the study guide,
Yeah.
please.
So yeah, so we talk about it. I talk about it a little bit here in this first one, which is really just let's see, where is that part? Yeah, I mean, it's just kind of this concept here that high levels of one hormone inhibits further release of another, if it's the hormonal stimulus, but it could be other things too. Let's look at this one here. The big one that we talked about that you did have to know the actual hypothalamic hormone was this thyroid hormone loop, right? So the thyroid hormone loop starts in the hypothalamus. Hypothalamus releases TRH, thyrotropin releasing hormone. You can just know the acronym, right? That TRH is released from the hypothalamus. It causes the anterior pituitary to release TSH, okay? And then that TSH immediately provides some negative feedback, right? The more TSH there is released, the less TRH there is released, right? And that makes sense because TRH's job is to tell the pituitary to release TSH. So if the hypothalamus sees that, oh yeah, we've got enough TSH, then there's no reason to keep releasing as much TRH, right? So that's kind of the whole idea of a negative feedback loop, is the result of that, or the result being that whatever it is that's released is kind of being sensed by the higher one, and it sees, okay, we got enough of that down there. We don't need to keep releasing this one at the bottom, right? So that would be a short feedback loop, right? But then we also have longer feedback loop where this TSH goes to the thyroid gland, tells the thyroid gland, does its job. I'm getting confused. Tells the thyroid gland to release thyroid hormones, right? The thyroid hormones do their job at the cells, but they also go up here and provide negative feedback in two places. The thyroid hormones tell the anterior pituitary to stop releasing so much TSH, because the TSH's job was to cause thyroid hormone release. So if we've got enough of that down there, then we don't need to keep releasing it. And then it also goes up to the hypothalamus and tells it to stop releasing TRH. Okay, and this is where this was an important one. We had that long hybrid question on it on the test for this section. This is where the goiter comes into play, right? Because the only thing you can see here, there's only one thing that gives the pituitary negative feedback, right? It's the thyroid hormones. Oops. The thyroid hormones are the only source of negative feedback for the release of TSH, okay? So if TRH is working, causing pituitary to release TSH, TSH is doing its job telling the thyroid gland to grow and release thyroid hormones. But if we don't have the nutrients that we need, being iodine, to make thyroid hormones, then there's no negative feedback, right? If we don't make the thyroid hormones, we block that negative feedback loop. So TSH is just going to keep pumping out. There's nothing to tell it to stop releasing, right? TRH will be inhibited by TSH in that short feedback loop, but this long feedback loop will not be stopped. And and so we'll get too much TSH release, causing the thyroid gland to grow and into that goiter thing. Okay. Does that cover your question?
Yes, thank you.
Yeah, for sure. Okay. Where else do we want to go?
Could we go over the vestibule, like the awareness of the head position?
Yeah, for sure. Some of my favorite stuff. Okay. So let's have some issues here. Oh, nice if I can actually show it to you on the pictures. So give me a sec so we can get this to work. Oh, it does this sometimes. It'd be faster on my computer here. Let me pull it up on there. Oh, that'll work. All right, got it. Okay, so let's go to this stuff here. Okay, so yeah, this idea of the semicircular canals being the ones that are sensing rotational movements of the head, right? So your head rotating one way or the other in that transverse plane of movement. And the idea here is that this whole idea of fluid lag, where when we first turn our head to the left, the structure of the semicircular canal, which contains this cupula thing that we talked about. Oops. Right, it's got this cupula, which has the little hair cells in it. Okay, that is attached to your skull, right? So when you turn to the left, that turns with it, right? So that turns to the left, but the fluid in there is going to go the opposite way. The fluid's going to go to the right because it's just going to go the opposite way of your head moving. And so that's going to move the cupula the other direction, right? And the whole thing about like having a piece of paper in front of you like we did and moving your head versus moving the paper is that when we turn our head to the right, in this case, what I'm doing, I'm turning my head to the right, the semicircular canals send the signal up to my brain that my head's moving to the right. So if I want to keep my eyes focused on that, then I need to move my eyes to the left, right, to stay there, right? But if I do that with the paper instead, I can't see, I can't focus on the paper because I don't have that input coming from my head that causes that reflexive movement of my eyes to keep them focused, right? So yeah, so we start with that fluid moving kind of the opposite way, okay? And that kind of, we went through this slide here showing that, right? This whole fluid lag idea. So the fluid moves the opposite way first, and then it kind of catches up. We get this constant rotation where the fluid is moving with the semicircular canals if we just keep spinning, right? But then when we stop spinning, now the canal and the cupula both stop, but the fluid is still moving, right? It's just going to keep going. So that fluid is moving around, and the cupula then moves back the opposite direction. And those things tell the brain different things. And then we kind of walk through this activity where we start rotation to the left, cupula and fluid movements go to the right, head movements obviously to the left. Continued rotation to the left, then it just kind of eventually will go, they'll all be moving together, so there's really no movement of the cupula. Okay, but then we stop rotation to the left. Fluid is still going to the left, but the head's not moving at all, so that moves the cupula to the left. And the brain has to interpret these sets of input differently, right? So So that's yeah, obviously very complex the way that the brain interprets that, but that's one piece of it, is coming from the semicircular canals. Also, obviously coming from other sensory input like the wind on your face and other sensory receptors that you're seeing, your eyes if your eyes are open, as well. So yeah, that kind of covers the semicircular canals part. Katie, did you want to talk about the vestibule as well?
Yeah, like just particularly like what's the difference between on the next slide? What's the difference between the turn forwards and the turn backwards?
Yeah. Okay. Sure. So we got these the special structures inside the vestibule are the macula, right? And they're or sorry, not the macula, the saccule, right? There's the saccule and the utricle, which are both forms of macula, but there's two different ones. And so the utricle is the one that is going to be parallel, positioned parallel with the ground if you're standing upright, and the saccule is perpendicular to the ground if you're standing upright. Right. So standing upright, the saccule has the weight of those little rocks, those little otoliths pulling down, so all the hair cells are bent to the kind of downwards in this case, right? So the input of the saccule being bent to the left and the utricle not being bent at all, that tells your brain that your head is upright, right? Your brain interprets that as the head being upright, okay? But then if we lean forwards, right, now we get the utricle ones bending down. The saccule is going to bend backwards, or not backwards, it's going to go back to straight up. And so the brain interprets that and knows that that is head forward, right? So if you had your eyes closed, you would still know that, right? And then the opposite way, if we bend backwards, now the utricle, instead of bending to the right, these ones are bending to the left now. Okay, so that's different input to the brain. Saccule is upside down, but it's really just going to make it go straight up, right? So the input from the saccule in these two is the same, right? But this one is different. The utricle is giving different input. One's being bent to the left, the other one's being bent to the right. So the brain interprets those two things differently, and it can tell you that your head is that way instead of this way. Does that help, Katie, or what else you want on that?
Yeah, thank you.
Yeah, for sure. Okay, what else? So I guess the other piece I'll just say on that, connecting the semicircular canals to the eyes, you would want to be able to do what we did in that hybrid question before, which was, like somebody was walking by you, and you're tracking that movement, and the whole connection of the head movement to the eye movement, and saying as your eyes are moving to the right, that's in your right eye, that's moving your abducens, or using your abducens to move your right eye to the laterally. And then that's going to activate the lateral rectus muscle, right? But in your left eye, it's using the oculomotor nerve to activate your medial rectus muscle, right? So that whole chain of events, connecting that would be an important one to use as well, or to know. All right, what else? We got time for one more if you want, or we can call it.
I have a question just about the hybrid that you were talking about.
Yeah.
So with that one, are your eyes moving, or is the object that your eyes are moving, or is the object moving?
Yeah, it's a good question. I was trying to remember how the hybrid was set up as I was just saying that. I don't remember exactly how the hybrid was set up, but you would need to know both ways. Yeah, yeah, you would need to kind of be able to interpret either example. Yeah. Okay. Anything else, guys? Good to see your pooch there, Trey. Cute. Glad you guys got to see Eve and Soren. Christian didn't make it in, but everybody else got to see the other two.
I was afraid to ask some questions because she was snoring in the back.
Yeah, that's funny.
So she was really loud.
Yeah, that's funny. All right, guys. Well, good to see you one last time, and I will be, I'll be available via email or, probably the best way if anything happens during the final and you just want to ask a quick question, just shoot me an email. I'll be on my email available for that because you guys will be using your cameras for the honor locks. You won't be able to get onto Zoom, but so good luck on the final, guys. Study hard, and good to see you guys. And hopefully I'll be praying for you as you're studying hard and thinking about all your other finals and everything else you got going. So, all right, guys. Good to see everyone. Bye-bye.
Thank you.
Thank you.
You're welcome.